Hello everyone, welcome back to another video, and the topic of today's video is virus. Now if you're from Earth, I'm pretty sure you've all encountered flu or common cold at least once in your life, or forget about common cold or flu. You've all heard about COVID-19, and we've all been affected by it in one way or another. So that is where virus comes in. Virus, when we think about virus, we think of these tiny little germs that we cannot see with our bare eyes, but they are everywhere. And even bacteria, we know that bacteria are so small that we also cannot see with our naked eyes. Even this bacteria will seem gigantic in front of this virus. You cannot see virus even with a standard microscope or a light microscope. You will need an electron microscope or a super resolution microscope. They are that small. So if we want to talk about some key characteristics of viruses, first comes acellular aspect of virus. So acellular means they are not made of any cells. So unlike human cells like us or bacteria, viruses do not have any cytoplasm or any cell membrane or even nucleus. They are just genetic material inside of a protein coat. So they are not made of any cell, they are acellular. Next is obligate intracellular parasite. Now we know parasite, we all know what parasite is. Parasite, in order to survive, they depend on other living organism to extract nutrition or to reproduce. They depend on others. Viruses are exactly like that. They attach themselves to a host cell or host organism, and they replicate inside that host cell in order to survive. So therefore they are called obligate intracellular parasite, and they are ultra small size. We already talked about that. They are around 12 to 300 nanometer in size. So we can say they are really small. Genetic material of viruses are either DNA or RNA. And antibiotics have no effect on a virus, because antibiotics we can already tell from the name that antibiotics will work against bacteria. So next time when you are taking antibiotics, be careful of whether you have viral infection or bacterial infection, because otherwise it will only lead to antibiotic resistance, which we will talk about in another video in details, because it is a very big problem right now. So be mindful of that. Now comes the big debate, which is, are viruses alive? Now this question, the answer to this question will vary from person to person. Some may say that viruses are alive, some may say they are non-living. Viruses do show some characteristic of living organism, like they evolve, they have genetic material, they can reproduce, but only inside a host cell. But viruses also lack the characteristic of living organisms, like they cannot independently, they do not have the ability for independent metabolism. So they exist in between, like a gray line between living and non-living. So for example, think of a USB drive, and which has a virus program in it. So when you have that USB drive itself, it's not doing anything. But as soon as you are inserting that USB drive into a computer, the virus will take over and spread. Now think of this computer as a host cell. The virus is entering the host cell or the computer, and they are replicating, right? So when viruses are outside of a host cell, they act like an inanimate object, but when they are inside a host cell, they act like a living organism. Now let's see what a virus actually looks like. So this is a very simple figure or structure of a virus, and in the middle of this figure we can see that there's the genetic material, which are DNA, they are either DNA or RNA. Now remember, a virus can only have one of this genetic material. It's going to be either a DNA or RNA. Both cannot exist at the same time in a virus. Now outside of this genetic material, or outside of DNA or RNA, we have this protein coat. You see this circular structure, this circular green structure. This is a protein coat, and it's called capsid. This is called a capsid, and this capsid is made of single protein molecules. You see this small circular round shapes, this green circular small shapes. They each are called capsomer. They are protein molecules that form this capsid. So each molecules are called capsomer. They are protein molecules. Okay. Now some viruses can have or may have an outer envelope. This envelope here, they are mainly made of a lipid or phospholipid. So they are mainly made up of lipid or phospholipids, right? Phospholipids. Now this envelope helps to keep the interior of the virus intact. Now outside of this envelope we can see some spike-like structures, this blue spike-like structures. These are called peplomer. Peplomer are glycoprotein spikes. So these are glycoprotein spikes. These spikes helps a virus to attach to a host cell. So this is basically what a virus looks like. This is the structure of a virus, basic structure of a virus. And virus can also be classified into two types based on the genetic material they have. So here we have DNA virus and RNA virus. You can already tell from the name that DNA virus will obviously have DNA, and RNA viruses have RNA as the genetic material. So here is a picture of a DNA virus, and inside the genetic material of this virus is DNA. And since DNA is more stable than RNA, they will mutate slower comparatively to RNA viruses. So they will mutate slowly, and also they're going to have long-lasting effects. A common example of a DNA virus can be the hepatitis B virus. So after that we have RNA virus. Now RNA viruses are really unpredictable because they mutate very fast, because RNA, as we know, is less stable than DNA, and so they mutate rapidly, and we can see that flus come back in new different form, right? This is because of RNA viruses. Here in this figure we can see that here is an RNA virus and the genetic material of this virus is an RNA, right? And the outer, we can see that the capsid outer layer, and inside of the capsid we have our capsomere. Coronavirus, in fact, is a RNA virus. And another fun fact of RNA virus is that in order to, it's comparatively harder to develop vaccines against RNA viruses. So up to this point we saw the characteristic of a virus, how they look like, and their classification based on the genetic material they have. So now we have to know about how they replicate inside a host cell, and they do it in 2 cycles. So we're going to go to that. So here as we can see, here are the 2 cycles that a virus use to replicate itself inside a host cell. But before that, we have to know what a bacteriophage is. So bacteriophage. Now you may wonder that we are talking about a virus here, so where did bacteria come from? Well, here phage, it's a Greek word and it means to eat. And bacteriophage is a virus that uses bacteria as a host cell to attach itself to the bacteria and insert its genetic material inside of that host bacteria and replicate itself. And therefore, that virus is called a bacteriophage. And this bacteriophage uses 2 replication cycle to replicate itself and reproduce. Now the first cycle that we're going to talk about is the lytic cycle. The first step of the lytic cycle is the attachment step. Now in this attachment step, a bacteriophage, this is our bacteriophage. This bacteriophage will attach to a host bacteria with a receptor site. Here we have a receptor site. It has binded to a receptor site of a host bacteria, and that is how the attachment step is conducted. So after the attachment step, when the bacteriophage has already attached to the host bacteria, it will then insert its genetic material inside of the bacterial cell. So the name of the step number 2 is penetration. Because it is penetrating its genetic material inside of that host bacterial cell. So this is the phage DNA. This is our phage DNA. And this is the bacterial DNA or the host DNA. So after the penetration of the genetic material of the bacteriophage, it will then move to the 3rd stage or step, which is replication step. Now in this step, what they're going to do is this phage DNA will use its promoter sequence to attract the RNA polymerase of the bacterial DNA. And with that RNA polymerase, they will make mRNA. So they will make mRNA, and we know that from mRNA we get proteins. So this protein will then, or enzyme will then disintegrate or destroy this bacterial DNA. So now they no longer, the bacteriophage or the phage DNA no longer has a competition with the bacterial DNA, and so they will slowly take over the host cell's machinery, and they will form new viral parts or virus parts. So that's where the fourth step comes, which is assemble. The fourth step is assemble. And here we can see that all the different virus parts have been formed. So here we have a tail, phage DNA, or a complete bacteriophage, and they will assemble together. In this step, or the fourth step, they will assemble together. And after that, in the last step, which is lysis, in this step, the bacteriophage, or in this step, the bacterial cell, they will burst open. So it's like puncturing a hole through the cell membrane of the host bacteria. And without a cell membrane is a very crucial part of a bacterial cell, and without this cell membrane, they will die. And as a result, they will release hundreds of new bacteriophage, and these new bacteriophages, they will then again go back to step 1 and then further attach to a new host bacteria. So this is how the cycle continues endlessly. And then we have another cycle which is called lysogenic cycle. Now in the case of lysogenic cycle, the first two steps are basically same as the lytic cycle. In the first step, attachment is going to take place. The bacteriophage is going to attach itself to host bacteria, and then it will insert or penetrate its genetic material inside of the bacterial cell. And then the fun part comes, which is the third stage for lysogenic cycle. And in this cycle, they will fuse their genetic material or the phage DNA with the bacterial DNA, and they will do that with the help of two enzymes, which are nuclease and integrase. The nuclease enzyme will cut a portion of the bacterial DNA, and with the help of the integrase enzyme, the phage DNA will fuse itself with the bacterial DNA, and as a result, we will get prophage. So this is a prophage. Here we can see that the phage DNA, the red portion, has fused itself with the bacterial DNA. This blue portion is our bacterial DNA, and they have fused together to form a prophage. So now the viral genome is inside of the bacterial DNA, and in the fourth step, here we can see multiply or division. So as the bacteria will now multiply itself, it will also make a copy of that viral genome. So every time they're multiplying, they're making a copy of that viral genome. Here you can see that we now have two of the prophage, right? So they are unknowingly spreading this virus. And this prophage can also sometimes go back to lytic cycle. So they can also go back to lytic cycle. So they will, what they're going to do is they're going to release themselves from the bacterial cytoplasm, and they're gonna come out and attach itself to another bacterial host. So we can see that we're again going back to step 1 here, right? So this is how the cycle continues endlessly. Now, after all this discussion, we may now think viruses as our enemies, right? Well, it's not entirely true, because viruses also have their own positive aspects. They play important role in medicine, in evolution, in science. Like viruses can be used in genetic therapy to treat diseases like cancer, and they are also helping to keep our ecosystem balanced. Like we can already see that they are controlling the population of bacteria. So that is how they are also helping in maintaining this balance of our ecosystem. So the next time when you are thinking of virus, don't just think of them as some tiny germs. Think of the big impact they have and the fascinating role they're playing in our world. So that is all for today's video, and stay tuned for the next video. Thank you.